Good afternoon. Welcome to the Bulkley Valley Research Center virtual seminar series. My name is Dawn Hansen. I'm the Executive Director, and I'm hosting today from the traditional unceded territory of the Gidimt'en Nation of the Gidimt'en clan, of the Wet'suwet'en Nation on the banks of the Bulkley River. Thanks so much to all of you for tuning in. We are always thrilled to host. We're always thrilled to see so many participants. I'd like to acknowledge the financial support of the province of BC through their gaming grants, as well as the Bulkley Valley Community Foundation and Woodsingqua Community Forest, and as well as our members. Our members are an important part of the center, and your membership and encouragement and participation is definitely key for the center. So thank you for that. Thrilled to host Lisa Wood today. As I mentioned, we have been working for a while to connect, and it's all lined up, which is wonderful. She's an assistant professor, ecosystem science and management at UNBC, applied plant biologist and dendroclimatologist, exploring influence of human-induced stress on forested plant communities. She specializes in forest silviculture disturbance types and studying the impacts of climate change on plants. Lisa's lab contains equipment and facilities for detailed analysis of tree ring records, quantification of cellular level plant anatomy, and investigation of plant chemical makeup. Her current focus is directed at elucidating the impacts of chemical herbicides on non-target vegetation populations in the boreal and subboreal forests of Canada. Thanks so much for joining us, Lisa, and welcome.
Thanks very much, Dawn. Welcome, everybody. Thanks for joining me this afternoon. As Dawn said, it is a bit chilly out, so I'm wrapped up in my blanket here in Prince George, and I'm speaking to you today from the traditional unceded territory of the Lheidli. Today, I'm going to give you a little overview of the current research that we've been conducting in what we call the plant lab at UNBC. I've had students working on glyphosate-based herbicides and their residues and persistence and effects of treatments for over 5 years in the plant lab so far. And we've been uncovering some interesting things that I'll walk you through today. I will provide a little bit of a warning that there are many graphs I will show you. I'll do my best to explain everything really well, but please feel free to ask at the end of the presentation. Any questions you have, I can return back to some of the information that I'm going to present. And I am excited to share some new results that my students have been working on. Very hard work going on in the labs, lots of people trying to wrap up theses and things right now. So an exciting time and lots of new information here. Okay. There we go. Don't do that. Okay. So there we go. I don't know how many of you are really familiar with glyphosate-based herbicides. I have been giving presentations about them for a number of years now, and so I try not to double up for those people who have seen some of my presentations in the past, but I do feel it necessary to do a little bit of an introduction on them for those people who don't really know a lot about the chemical itself. Of course, these are herbicides that are used for a number of reasons to remove vegetation from different environments. We investigate them largely in forested ecosystems, so more natural settings, but in forest cut blocks, they're used primarily to remove competing vegetation from around crop trees. And so we are studying what happens when that the chemical is used in that way. But of course, they're also used in agricultural settings, in maintaining right-of-ways for hydro and oil and gas pipelines, well sites, along roadways for sight lines, and all sorts of other applications. It was previously thought that the half-life of glyphosate was quite short. A lot of the historical research on the use of glyphosate suggested that glyphosate could be degraded, at least in its half-life, within less than 1 day. Some of that research took place in warm climates and some in laboratories at room temperature. And so the effects of glyphosate were studied in very certain environments. It has been since discovered that glyphosate can persist longer in plants, water, and soil than was previously recognized. And this is just really, I think, because of glyphosate becoming more broadly used across the world, the extent of its use increasing, really leading to the necessity of more research on glyphosate and more research in different types of environments. And so researchers are realizing that the half-life of glyphosate is highly dependent on what environment you study it in. Okay. So it's a little bit difficult to make generalizations across a number of different environments for this reason. But in our forests, in northern British Columbia, we find that the persistence, at least in plant tissue, which is what we specifically look at in my lab, is quite a bit longer than was originally thought. We see persistence for at least a year at levels that are still sometimes above what is considered to be an adverse effect limit or a minimum residue limit. So here's just some of the early research that I conducted. This was published in the Canadian Journal of Forest Research in 2019, just looking at what the residue levels were like in different plants. These were actually from an area around Tumbler Ridge, British Columbia. And you can see that the white dots are the amount of glyphosate residue, and they go up to about 4 parts per million. Average is about 1 part per million here. And this is a whole year after application has taken place. So that is higher than would have been previously thought, even though it seems like it's potentially still a low level. So we generally acknowledge that, yes, okay, glyphosate is present in these forest ecosystems, maybe in soil, maybe in water, maybe in plants, longer than we thought. But the real question is, what is it doing there? Is it causing a problem? Is it bad that it's there? So this has really been the focus of the research in the plant lab, is to try to pick that apart a little bit. And the first thing that we observe when we walk into forest ecosystems years after applications take place is the change in the vegetation. Of course, we can expect the removal of some vegetation. The whole point of the treatment was to target competing vegetation. So we noticed that the trees and the larger shrubs that were in direct competition with the conifers that are being planted in these spaces are usually dead or significantly impacted by the treatment. So just that in itself, the removal of a large amount of vegetation is fairly impactful. But what we wanted to know is, in the plants that remain, is there an impact? And the plants that remain are quite often impacted because they actually did get a little bit of the treatment, right? So quite often glyphosate is applied in what we would call a broadcast application method. So a plane or a helicopter has been flying over and released the spray, or somebody's walking around with a backpack and a wand and not really being very discriminatory about where it's sprayed. They're just spraying across the space in order to get good coverage. And when that happens, the plants that are targeted, that are larger, are receiving the full concentration of the treatment, and they're dying. And then the plants that are underneath those or beside those are getting a little bit of the off spray. And when that happens, these plants don't die, but they get stressed out by that application. And so this is where we see the residue persist in tissues in plants that really haven't died from the treatment. So they're hanging on to it, and they're passing the chemical into the shoots, into the roots, and all through the systems as they grow and change throughout seasons. So here I'm showing you a picture of some of this effect to what we call an off-target plant. On the left-hand side, we have a fireweed plant. So right here, the black and white diagram of the fireweed is what we would normally expect a fireweed plant to look like. Probably you've all seen them. So usually just a single erect stem with sort of large lanceolate shaped leaves that come off the main stem. And in the greenhouse, we sprayed this for fireweed plant to see how it would grow, to see if we could replicate these effects. And you can see that, in fact, it no longer has one erect stem. It sort of forms this strange bunching effect. So we get lots of branching, and instead of having larger lanceolate leaves, you can see there is still some larger leaves, but in the new growing material, the new tissues, we see these clusters of very small bunched up leaves. So why is that significant? Is that important? Well, if you think about habitat requirements for certain insects or invertebrates, if you think about forage requirements for some animals, leaves like this could be less attractive or maybe more attractive. Forage may be less attractive. Habitat, probably species dependent. So some species might really like these little clusters of leaves that are being created, and it might create new habitat. Other insects or invertebrates that requires maybe a larger leaf surface to land on for mating ground or foraging ground may not like these small leaf bunches. So yes, there could be an impact here from these changes across, and we're thinking across the whole block, right? Or across many different forest cut blocks. Similarly, in other species, we see the same sort of response. So we don't always see similar responses across species, and I'll get to that later. But in this particular way, we do. We see that, again, this is a raspberry plant on the right. Here's a diagram of what we would normally expect a raspberry plant to look like. I'm sure most people here know what raspberries look like, but we have these leaflets that have 3 to 5 parts to them in the leaf. And then here again, we see these very small little clustered bunched leaves, which really don't have a lot of surface area. It's problematic for the plant because the plant needs leaf surface area for photosynthesis, so it can't produce the food for itself that it needs to produce with small leaves like this. So it's really struggling to get back to normal. And because it has low energy, there are other consequences for it, which I'll speak to in a second. Sorry. Okay, so here's some more changes to leaf morphology. This particular research was conducted by Catherine Timms in 2020, published in Global Ecology and Conservation. Changes again to raspberry leaves. You can see they're not quite as small and bunchy, but they're very, very chlorotic, so not green. There's definitely an interruption in photosynthesis happening here. And then in cranberry, high bush cranberry leaves, we see some deformation, some asymmetry, which is known to be challenging for foragers. Animals like symmetry in their leaves, and just some strange curling and browned leaf effects. So different species having potentially different responses in terms of their ability to photosynthesize post-application. We also see anatomical changes to flowers. This work was conducted by Alexandra Golt in 2021. She was working on an undergraduate thesis during this time. This was published in Frontiers of Plant Science, and you can see that there's changes when she looks at, this is Rosa sicularis, so prickly rose. When she looks at rose petal color, the shape and condition of the buds, this looks like a really old bud, but these are actually brand new buds, but just from treated areas compared to what some buds would look like in control areas. Similarly, in the anthers of the flower, these again look like they're very old anthers, but actually some of these, this is what a treated anthers would look like, and untreated anthers would be somewhat more healthy looking, nice bright yellow color. So even in buds that are just being opened up for the first time, we have this strange effect from glyphosate treatment. Okay, so why is that important? Changes in color, of course, are important for pollinator species, and pollen is really important for fruit production. Right? Without pollen, we can't produce fruit, and fruit is a very important source of food for many animals, and the seeds inside the fruit are also very important food for many animals, and also really important for the plants themselves, right? They can't sexually reproduce without fruit and seeds. So without that pollen, it's difficult. Pollen was significantly reduced in treated areas, so not viable. It's there, but it can't germinate. So this really cool picture taken by Ellie Gold shows the pollen tube that's formed in a healthy pollen grain. After it germinates, this pollen tube grows down into the flower and allows the sperm to fertilize the ovule. Without that pollen tube, we don't get pollination, and in non-viable pollen, we don't get the production of the pollen tube. So here we see in cut blocks, 1 year after treatment, we do not have the same amount of viable pollen, and this is in Rosa secularis over here. And then on the left, this is new work where we're expanding on this, looking at some other species, and Ali is writing this up for her master's thesis. And she studied mostly fireweed for her master's thesis. And in fireweed, we saw the same trend in 1 year post-treatment areas. The pollen is significantly less viable than in control areas. However, by 2 years post-treatment, there's no significant difference in pollen viability any longer. So a little bit of a success story. We do see an effect, and it is for about 1 year here, but by year 2, potentially we have some recovery in the ability for pollen to become viable again. So that's a good news there. However, for that 1 year or potentially 2 year period of time, the plants that are remaining in these spaces are not able to produce the same amount of fruit and food as they would normally. So this leads us to thinking about fruit production. We really wanted to go, we're assuming that pollen viability means something for fruit production, but we wanted to make sure that that checked out. So we went and we measured fruit production. And again, for Ali's thesis, her master's thesis, she surveyed some fleshy fruit producing plants in our forest. So on the right-hand side here, the different species that fell in the plots we measured. And in out of these areas, the control locations, about 91% of the plants we investigated were producing fruit. And in the sprayed areas, so 1 year post-spray, 99% of the plants were not producing fruit. So very stark contrast there. Again, pointing back to the plant's ability to really get energy for itself and photosynthesize, right? If a plant can't photosynthesize properly and produce enough energy, it's never going to get to its reproductive stage. And if it really does manage to get to its reproductive stage, the pollen is likely not viable. So in these conditions, it's very difficult for a plant to actually get to be able to produce fruit in a cut block 1 year after it's been treated. Okay, what other interactions have we seen? So glyphosate is a natural chelating agent, which means it likes to bind to heavy metals. It also is a chemical itself, so suggesting that when you introduce a chemical into a system, other chemicals are going to respond to that chemical. Things are going to change a bit. So we wanted to see if we could figure out a little bit about that. This is an example of what the glyphosate molecule looks like. We have 3 different chemical groups here, and these groups can provide binding sites for the metals in the chelation process. Previous research pointed us in this direction. So originally glyphosate was used as a chelating agent before it was ever discovered as a herbicide. So there's previous research, so CACMAC being one of the researchers in 2009, who was investigating agricultural plants and the effects of glyphosate in terms of the mineral response in the plants. And we can see here, just for example, when they apply different rates of glyphosate. So here we go from no glyphosate being applied to 1.2, I would assume, it's percent of recommended, so a higher concentration. We get a decrease in magnesium content in the plant. This is an example from soybeans. We also see some other trends fall out. And so based on the trends we had seen, or the minerals and metals that were important in previous studies, one of my students, Nicole Botten, who has since completed her thesis, also decided to investigate some of these minerals, but in operational spaces in our forests. And so in 2022, she produced a thesis. This is yet to be published. But she found a similar trend in magnesium to what CACMAC found across plants. So fireweed, red ochre dogwood, and willow, we see a consistent decrease in the amount of magnesium produced. And this is up to 12 years after applications take place. We also found a decrease in nickel across all of the plants and years investigated. You can see that some of the other metals and nutrients that were investigated didn't really have a consistent trend. So we see maybe mostly an increase in zinc, but a few instances where it decreased, and then really no particular trend in manganese, iron, or calcium. In magnesium and nickel, where we see these decreases, I mentioned it can be up to, Nicole investigated up to 12 years after applications took place. And so that's a fairly significant period of time. I should mention that at 12 years, we really see very little glyphosate residue left. Nicole did find that only in 2% of samples was glyphosate still present, and it was only in roots of a particular species. And in those roots, it was a very, very low concentration. So this tells us also something interesting. It tells us that even though the glyphosate residue itself has actually degraded and gone away, the effects of its initial presence potentially could be felt after the residue is gone. So these changes to mineral nutrition or mineral presence and absence have actually persisted up to the 12-year mark, regardless of the concentration of the residue that's present there. So we're not really sure how long these systems take to recover. They obviously take different periods of time to recover depending on the way they've been affected. So minerals taking maybe a bit longer to recover back to normal than pollen, for example, or pollen viability. What about other plant chemistry? So we know that plants contain all sorts of chemicals, some of which are used by, lots of which are used by humans for various purposes. Berries, of course, being a very good source of antioxidants. So we wanted to know a little bit about how glyphosate might be affecting the internal chemistry of plants. So Catherine Timms, in her study, she looked at the leaf deformation, but she also looked at a chemical called rutin, which is a flavonoid in a plant. It is a prevalent antioxidant, so it's one of those chemicals that make fruit a good antioxidant for us. And in raspberries and cranberries, she wanted to see if there was an impact from the treatment of glyphosate. We studied these in some field trial plots we established at our Eliza Lake Research Forest. And you can see on the right here that it's not an amazing result, but we do see in raspberries that the untreated, so the control plants, have a generally higher amounts of rutin than the treated plants, especially at week 2 after spray. So this is just within the same year of spray. So we measured weeks after. We seem to have sort of a flush of rutin in week 2 that we don't see in the treated plants as significantly. Amounts of secondary metabolites like rutin that are produced by plants are reflective of what's going on in their environment quite often. So a great sunny, warm day might induce more production or less production than a rainy day, or other types of environmental factors like that. So we could potentially see that maybe there's a difference in week 2 because something in the environment was causing a increase in rutin in the control that didn't happen in the treatment. That's one of the possible explanations for that. However, interestingly, in cranberry, we see the complete opposite trend. We see that the treated cranberry plants actually have more rutin than the untreated cranberry plants. And so this is just one of the very common things we see with glyphosate when we're talking about plant chemistry. Plant chemistry is pretty complex. And as humans, I think we really take for granted the fact that these are all different species. We like to just say, oh, they're plants, all the forest plants. But this would be like trying to compare an iguana to a howler monkey. They're completely different species. Yes, they belong in the same kingdom, but they are totally different plants. And so when we're comparing plant to plant, it's very hard to generalize. And so seeing differences in responses is really not that surprising. It just makes our job as researchers to try to tease out what this all means difficult. However, having said that, what we can generally see is across most of the research that we've conducted, there is an effect. So there is glyphosate is inducing a change. We're not sure always if that change is going to be positive, negative, or neutral in terms of its overall effect. We have looked at amino acids. This is some very recent data. I'm working on this project with, we collected these flowers and been working on some of this data with Ali Goltz and also Brian Hampton, one of my undergraduate students. And we collected fireweed flowers and stream violet flowers, and we sent them out for amino acid analysis. We analyzed 15 different amino acids in these flowers. Because amino acids are a really important thing for animals, right? Animals get a lot of their amino acids from plants, especially, of course, herbivores. And pollen can also be a very, very important food source for a lot of different insects. Bees get their complete diet, essentially, most bees anyway, from pollen. They, of course, eat nectar as well, but they're really only getting sugar from the nectar. So pollen is a really important food source, and pollen is very high in amino acids and found in flowers. So we tested flowers in fireweed flowers. 13 out of 15 amino acids were significantly lower than in control samples when the glyphosate had been applied. So that's a pretty significant effect. However, again, we see a little bit of an opposite effect in the stream violet. In stream violet, only 3 of the 15 amino acids were significantly different, and those 3 amino acids were actually higher in the treated stream violet flowers. So differences here could be that stream violet is a very low-lying shrub, herbaceous plant, and it was also phenologically in a little bit of a different place than the fireweed when we sampled it. The fireweed was quite obviously more affected as it's a bit higher, so probably more exposed to the spray than the stream violet. And the stream violet was already sort of not decaying, but started at the end of its reproductive cycle when we were sampling, versus fireweed flowers, which were more highly in bloom at the particular period of time that we sampled. So potentially a little bit of confounding effects there. However, I can say that this effect with fireweed flowers being consistently lower in the 13 amino acids that we measured out of the 15 is a pretty significant result there. Did I skip 2 there? Yes, I did. Okay. So another piece of our research has been looking at the effects of the environment and how it interacts with glyphosate's presence and effects in the ecosystem. So this was spurred on by some research by Nicole Botten. So this was part of the research published in Forest Ecology and Management in 2021. And she is the one who discovered samples 12 years after glyphosate applications that still had a bit of residue. So you can see here we've got those 2% of samples with glyphosate present in year 12. However, the interesting thing, and the reason I'm showing you this, is actually because of the difference between the number of samples and the concentration. Also, it's not shown in this graph, but the concentration follows the same trends. So the number of samples and concentration of these samples is containing glyphosate after spray is higher for longer in the BWBS zone compared to the SBS zone. And these are our biogeoclimatic zones. So in our boreal white and black spruce biogeoclimatic zone, we see that 96% of samples 1 year after glyphosate applications contain glyphosate residue. And they actually contain fairly, it's low concentrations in terms of government regulation, but it's higher concentrations than we kind of expect to be there a whole year after. And then you can see the trajectory of degradation here in the BWBS. By 3 years post, we have 66% of samples that still have glyphosate. By year 6, we have almost 50% of samples still have glyphosate, which is quite a surprising finding. And then down to 2% by 12 years. In the SBS, or subboreal spruce zone, closer to Prince George, we see that there's this exponential decrease in the first 3 years. So by year 3, 27% of samples contain glyphosate, and then by year 6, only 5%. So it's this difference in trajectory that we're really curious about, the BWBS zone being much colder with longer winters than the SBS zone. So our first thought, of course, going to, okay, does temperature have an impact on how glyphosate degrades or persists in the environment? So I have a student, Hariharan Sendamangalam Varudaraju, who is looking at this exact effect. And he conducted a number of isolated and controlled experiments for us so that we could really get to the bottom of the issue. We decided to target strawberry plants as they are a common food source in our forests, especially an earlier spring food source, berries being produced quite a bit earlier than other plants. And so we obtained some wild strawberry plants and we grew them in greenhouses at different temperatures, some treated and some control plants, of course, at different temperatures and also different photo periods. And so he's writing this up for his master's thesis. And we did find some interesting things. This is just one little snippet of his data, but you can see here that these first 2 bars are plants that are grown at 8 degrees Celsius as an average temperature. This middle 2 are plants that are grown at 12 degrees Celsius. And these last 2 bars are plants that are grown at 20 degrees Celsius as an average temperature. And you might think, oh, 8 degrees Celsius is pretty cold for an average temperature, but think about the north in the spring. It's actually not that uncommon. So we see here that the plants treated with glyphosate, which are these lower bars or the second bar in each pair, in the 8 degrees and 12 degrees Celsius chambers, the glyphosate treated plants were not able to photosynthesize as well as the control plants. So this is a measure of their photosynthetic ability. And you can see here their photosynthesizing or their photosynthetic capacity is a lot lower than the control plants. And interestingly, when we get to 20 degrees Celsius, the glyphosate treated plants, there's no difference in how they can photosynthesize compared to the control plants. So this starts to tell us a little bit about where temperature might be playing a role in the plant's response and the plant's ability to maybe tolerate or process or degrade glyphosate and then still be able to get on with its life and not be too stressed out. So there's definitely something going on there with temperature. So we're very excited about these results. And I'm sure Hari is looking forward to publishing this. Okay. Did I do it again? Oh no, I didn't. Okay, sorry. So all of these things that are going on with plants are pretty significant because, of course, plants form the bottom of our food chain. So I started to talk a little bit about habitat quality and forage quality. Here, we started to ask some more questions about that. And what we thought about was plants, we know there's an effect to the anthers of the plants, the flower color, and the pollen quality. Well, we know that bees and some other types of invertebrates might see pollen in different ways or see flowers in a different way than animals and humans see them. And there was this past research done by a Japanese researcher who photographed rose flowers and I think a bunch of other species as well, and really tried to show what insects might see, and especially bees might see in a flower compared to what the human eye sees. And you can see here in the rose, the anthers are really bright and fluorescing compared to we see yellow and pink. And so this is a way for foragers to find flowers and food, right? To find the food they're looking for. So we tried to think about how change in color and change in pollen might actually lead to a change in attractiveness of flowers for different species who need to forage on them. This was an image that Alexander Golt took of Rosa acicularis flower under what we're using for our fluorescent light, which is a royal blue light. And so here's some further images of that. Here's some anthers from fireweed that are photographed under that royal blue light on the bottom. And you can see how they're fluorescing so beautifully compared to just a regular white light, what they look like under the white light of the microscope. There's this bright green from the pollen and these bright orange anthers that I think must be very attractive for the insects. However, we wanted to know if in fact insects or bees specifically were more or less attracted to these changes. So as again, part of Ali's master's thesis, she is looking at the fluorescence of fireweed and its attractiveness to bees. So are they visiting the flowers in the same way that they would normally have? And in fact, in the fluorescent images, Ali did find that the average blue pixel intensity is significantly reduced in treated samples. So what that means is that under certain conditions, there is less blue reflectance happening. And so bees who actually prefer the blue, they're more attracted to the blue light, according to some of the literature that's out there. This could potentially be problematic, but yet to be determined. We're still connecting those dots. We are looking at changes in general insect and invertebrate populations. And one of the species that we've specifically been looking at, Ali's been looking at Bombus species, different Bombus species in forest cut blocks. She found that there were significantly fewer Bombus bees found up to 2 years post applications in her sites. And this is a little bit of an overwhelming graph, but I'll try to break it down for you. She did see that there was more than just a reduction in bees, and not always a complete reduction, but actually more importantly, a community shift in the species that are present in treated areas compared to untreated areas. So what we have here is you'll see that there's some purple or pink, however your eyes see that color. There's some that say control. So those are our control blocks. And there's some that say 2020. So those are 1 year after treatment, and some that say 2019. So that's 2 years after treatment of glyphosate. And then we have our Bombus species that are interacting in here in blue. And so some of these species are going to be more associated with our control blocks, and some are more associated with our treated blocks. And some are more associated with treated blocks 1 year after treatment, and some are associated with cut blocks 2 years after treatment. So, for example, Bombus sandersonii here was more likely to be associated with control areas or blocks treated in 2019, which were incidentally more similar to the control blocks than blocks that were treated in 2020. Conversely, Bombus vagans, another species of bumblebee, was more associated with blocks that were treated 1 year after. So this just tells us that there's a change in species community composition of insects and of specifically Bombus species here. There's a number of theories of why that might be. Ali has been researching a lot about bee behavior and their colony establishment, and so there's some theories about nesting requirements and, you know, what does the queen know before she goes dormant for the winter? Well, the blocks have just been sprayed, so there's probably like a lot of nice vegetation on a block. It comes and gets sprayed, but it doesn't die right away. So potentially the queen doesn't know that this is a degraded area or an area that has been sprayed. She's setting up a nest somewhere that she thinks there's a lot of food, and then perhaps she wakes up the next spring and all the vegetation there is dead, but there's still a lot of bees now in the area that she has just built a nest for. So that may be one of the theories. Another theory could be that open spaces that are fairly vegetation free actually provide just corridors for flying. Bombus species are one of the species that can fly a bit longer than some other types of bees, and definitely longer flight paths than other flying insects. So it's possible that they're using cut blocks as corridors, and so we're capturing them because they're just moving through the space. So a number of different things that could mean. This is brand spanking new data that I just put together last night, so it's a little bit unfiltered here, but this is showing just some other invertebrate communities that we've measured in treated cut blocks compared to controls. In species like Collembola, so those are our springtails, we see an increase in treated areas. Also in the Thysanthoptera, we see an increase in treated areas. These are species, thrips, and springtails are species that like to feed on dead leaf material. So that's not a huge surprise that we might see more of them being present in treated areas. There's also some decreases, so Diptera, so our flies, so if you definitely a difference in those and some other species that we're still trying to pick this apart. So again, pointing towards this idea of a community shift. So I'd like to introduce that as an idea being something that we should be thinking about over just biodiversity. You hear people talk about biodiversity a lot, and you hear people say, oh, are there changes in biodiversity? Because of course, biodiversity is an important factor, but actually community composition can be just as important. And if we're seeing a shift in community composition, it may not always equate to a change in biodiversity, because you might have the same number of species, you just have different species. So it can actually change the way an ecosystem functions, but we don't see that if we only measure biodiversity. So I think that's really something important that we should keep in mind. Lastly, I'd just like to touch on the fact that we've been looking a little bit at soil and fungal communities in soil. So we started to think about, if we're seeing effects to plants, probably there's something going on with all these mycorrhizae that are connected to the plants under the soil. And we know that residue can persist in soil for a little bit longer as well in colder areas than it originally was thought. So because of these associations, is there an effect to fungi? And so I had a undergraduate student, Helga Haller-Busch, working on this project, just doing a preliminary stab. So we established some field plots at Eliza Lake Research Forest, and we measured some fungal traps that had been set in these plots to see if there was differences in biomass of the fungus. And we also took some soil samples. And the soil, I just put this up here to show, the soil of a lot of these plots was very sandy. And so this led us to one of our very interesting findings from this work, which was that all our control plots that were more than 10 meters, well, actually almost exactly 10 meters, because we laid them out as such, 10 meters away from our treated plots. So 10 meters in between. So the control samples potentially more than 10 meters away. All the control samples contained glyphosate, which was very unexpected. So all of our samples that we collected of the soil, both from the control plots and the treated plots, ended up containing glyphosate. So this has led us to a very interesting research question about whether or not, or about how glyphosate moves through sandy soils. And we used that 10-meter buffer between plots because that's the buffer that's left in a lot of riparian areas for spraying. Don't spray 10 meters from a stream. So we were wondering if in sandy soil conditions that 10-meter buffer is really going to be effective. So we're going to be diving into that in the future. So what did Helga find? She found that across the plots, her hyphal biomass, so the amount of fungal hyphae found in these traps actually increased with the presence of more and more residue. So it was the opposite of what we expected. Although once we dug into literature, we were happy to be able to explain the fact that we think that a lot of some types of saprophytic fungi and potentially pathogenic fungi actually really like glyphosate because glyphosate can be tolerated by some fungi and then not tolerated by others. So it basically gives some fungi a competitive advantage and allows them to take over the environment. And so for seedlings and crop trees, maybe not a great thing because actually you can be increasing the number of fungi that can negatively affect crop trees. So something that maybe foresters might want to be aware of and look into, but other plants as well. So pathogenic fungi can also be, have negative impacts on some other plant life that are present, and it would discourage the production of potentially helpful mycorrhizal fungi that these plants need to grow. So we're expanding on that study now by doing some genetic analysis of the soil so we can actually tell what species are there so that we can confirm our theory. Okay, last slides here. I just wanted to tie this up to the management perspective, and I know I'm over time, so I'll really try to swing through this. But as I mentioned at the very beginning, glyphosate-based herbicides are used in a lot of different ways in British Columbia and across the world. These are just some of the ways that they're used. And I have to say that actually, since I've been starting to do this research over the last 5 years, the response from the forest industry has been very positive. They really have made an effort to dive into this research and think about it. And I have talked to a number of licensees that are decreasing their applications or completely eliminating their applications of glyphosate because of some of this research and because of some other issues, of course, as well. And I would say that really our big user of glyphosate-based herbicides is agriculture. And in agricultural situations, across the entire world, the use of herbicide in general has increased very significantly. So this data is from the Food and Agriculture Organization of United Nations, and in the world, about 90%, between 80 and 90% of what we apply in terms of herbicide is actually glyphosate. So you can imagine these trend lines are probably pretty similar for glyphosate. The actual concrete amount might be a little less. But agriculture is our big user, and they are not required to leave the same kind of buffer zones around their streams and waterways as other industries are, and they apply many times a year. So it's definitely probably somewhere that we need to think a little bit. Here's what our use in forestry looks like. This is just from 2016, so I didn't have a more recent graph, but you can see that our amount of use annually has decreased a little bit. And I would say this is probably continuing to decline in the last 5 years. Okay, that's all. I hope somebody has a question for me. I didn't put my thank yous up on this slide, but there's a lot of people that have supported this research. West Fraser, Canfor, BCTS, the 3 major licensees in this region actually have been supporting this research. The Habitat Conservation Trust Foundation have been a good supporter. The Weston Family Foundation, NSERC has supported lots of the research, and many others. So thanks to everyone for their support. And this is my contact information if you would like to get in touch with me.
Thanks so much, Lisa. It's fabulous information.